you have exhausted your glutathione levels. Just 1 minute, 1 minute, 1 So if, because of other reasons, you have depleted sources of glutathione, for sure, you will get paracetamol toxicity. So this glutathione for further metabolism to non-toxic metabolite. 75% if you have, and that can result in 70% depletion of the glutathione. So you do require the glutathione, even as you do your pharmacology. Glutathione is very, very important. It even helps to protect quite a number of cells from undergoing hemolysis. So glutathione levels in the body are very, very, very important because they protect quite a lot of things. They mop up, it helps in detoxification of electrophiles found in the body through metabolism of drugs, because glutathione in itself is a nucleophile and can mop up the very toxic electrophiles. So we look at now that metabolism of paracetamol. So we are saying you can have two routes of metabolism. You can see we are saying you can have sulfate conjugation as a major route, or you can have glucuronic conjugation, and those are the two safe. Those are two safe methods of not having a toxicity. If we did mention phenacetin, and we did mention if you take phenacetin, it can also be metabolized to acetaminophen through oxidative N dealkylation. That's an ether group oxidative N dealkylation, and that's a major route, will give us acetaminophen. But minor route can also be hydrolysis of the acetamide group to give us to give us the derivative, the amino derivative of the ethoxy derivative, to give us the amino derivative, or the aniline ethoxy derivative, aniline ethoxy derivative, and this aniline ethoxy derivative can be responsible for methemoglobinemia and even hemolytic anemia. Responsible for methemoglobinemia is where the Fe2 plus of the hemoglobin has been oxidized to Fe3 plus, and you know Fe3 plus cannot transport oxygen. That's what we're calling methemoglobinemia and even hemolytic anemia. Then also, we did mention acetanilide, and we did mention it is not a drug. It cannot be used as a drug again because of its major toxicity. Acetanilide, which is just the acetanilide group there, or acetylamide group there. It can be metabolized major to paracetamol, acetaminophen through oxidative hydroxylation, parahydroxylation. But again, it can be in a minor route, again, be metabolized through hydrolysis to, again, aniline itself. And again, aniline is known, it's very toxic, methemoglobinemia and hemolytic anemia. And that is why, first and foremost, we ruled out the use of those two drugs. Use of course, the phenacetin, this is converted to phenacetin, then the phenacetin, as we had said, as we are going to see also with paracetamol, because of this NH group, that N, you see this has a hydrogen. The hydrogen, remember this is Bn, C double bond O CH3, NH, C double bond O CH3. That hydrogen, again, can allow for oxidative hydroxylation to give us the hydroxyamine. This is the hydroxyamine derivative. And this hydroxyamine derivative is hepatotoxic. And this is what becomes hepatotoxic. It is the same group there. And then it can also undergo hydrolysis of that, again, to give us this group compound, which is still hepatotoxic, because all that it does is that it can give us this group. So we are saying phenacetin can come up to there and give us also the N-acetyl iminoquinone. The acetaminophen or paracetamol, that is its structure. Again, because of this group here. Remember, it has a hydrogen here. This is the rare route of metabolism, which is being catalyzed by cytochrome P450 enzymes, as we have said, the E1 and the 3A4. So that can metabolize, those two enzymes can metabolize the degradation or metabolism of acetaminophen, and this will give us this group here. So you can see that's a double bond. It will go through this first. This the one is which is this one. So it will first go to this step, but of course without the C2H. So it will actually go through this step. This is the right step. So it will first of all go to this acetaminophen, will go to this step first and then go to this. But ultimately it will go to this. And this is the metabolite which we are calling the N-acetyl iminoquinone. And this is a toxic, very, very toxic metabolite. But we did say if you have enough supplies of glutathione, TSH, and what is glutathione made of? So that at least I need a bit of interaction from you. What is glutathione made of? Can I get some answers? What is glutathione made of? I hope you have done metabolism. I think normally with Dr. Kegundu. Have you done metabolism? I hope you have. What is glutathione made of? Glutathione, quickly, let me get more answers, please. And what are the main metabolites on glutathione conjugation? What are the main metabolites, or what is the main metabolite? What's the main metabolite on glutathione conjugation? Main metabolite on glutathione conjugation? What is the main metabolite on glutathione conjugation? Main metabolite on glutathione conjugation? Main metabolite on glutathione conjugation? Yes, we are getting the answer there. Glutathione is made of glycine, cysteine, and glutamic acid. And once it binds an electrophile, there is systematic degradation or removal of the amino acids. So what is usually the metabolite? What is the main metabolite of glutathione conjugation? What's the main metabolite of glutathione conjugation? What's the main metabolite of glutathione conjugation? These are things you have done, I believe. It should be the three enzyme, not the three amino acids, demopractitia, not 2. There has been glycine, cysteine, and glutamic acid. What is the main metabolite of glutathione conjugation? As you think about that, so what we are saying is that the glutathione helps a lot, and the glutathione will come and join this very highly toxic metabolite. It's an electrophile, it has electron deficiency. So glutathione has a site through its cysteine. It has cysteine, and you know cysteine as an SH, or what you call the sulfhydryl groups. Cysteine has sulfhydryl groups, and through that sulfhydryl, you know, sulfur has 2 lone pairs of electrons. Through those lone pairs of electrons, they will bind these electrons and neutralize it. But of course, this can then further break down, you know, glutathione will first lose its glutamic acid, and then after that lose its glycine, and the final product is usually the toxic metabolite plus cysteine, or what we call the mercaptopuric acid. So renal excretion as mercaptopuric acid or cysteine conjugate. So these are things that I believe you have done. You must have done them in your metabolism. So if this does not happen, and this can fail to happen if you do not have enough glutathione, you might have low levels of glutathione because of you are malnourished. If you're malnourished, you might have low levels of glutathione, because you can see these are amino acids. You can have low levels of glutathione because if you are using certain drugs, or if you are using alcohol on chronic basis. If you're using not alcohol, but if you are mainly if you are malnourished, you can have low levels of glutathione. Actually, if you are malnourished, you can have also. So that is one reason why you can have this pathway not happening, that you are malnourished and therefore you do not have enough glutathione levels. If you are using alcohol on chronic basis, you find that there is induction of the cytochrome P450s, and therefore this route, more of the toxic metabolite is formed. That is when you use, I have said, if you cytochrome P450 enzymes, which are responsible for formation of the toxic metabolite. So you will have very high levels of the toxic metabolite, and once you have very high levels of the toxic metabolite, then you will exhaust, or you will deplete your available glutathione, and therefore you lead to toxicity. What we are therefore saying, if this metabolite, the N-acetyl iminoquinone, is not mopped up, then it will go and bind your sulfhydro enzyme, your sulfhydro proteins, and if the sulfhydro proteins means those are cellular structures, it will bind your cellular structures or your structural proteins within the kidneys, within the liver, and it can result into a lot of toxicity. And that is how those who commit suicide by taking paracetamol in very high doses, they die of hepatotoxicity because of the high levels of the iminoquinone which are formed, and they cannot be neutralized by the glutathione. They exceed the glutathione stores, and therefore the patient or the person dies of hepatotoxicity. So no drug is safe, none at all. That's why we stress that each and every drug must be taken at the right dosage, even paracetamol. So what's the mechanism of toxicity is what we have already said, that paracetamol normally undergoes sulfate conjugation and glucuronic conjugation. If you do have the cytochrome P450, and if it is acted on, it fails to go through that main route of metabolism, and it goes through the minor route of metabolism, which is the cytochrome P450 oxidation. It will give us toxic intermediate metabolite, which will bind with sulfhydryl enzymes or cellular proteins to give us, or this one is binding with this GSH, all that one will give us non-toxic metabolites. If we do not have enough of GSH, then that's where it will bind our cellular tissues, it will bind our enzyme, resulting into death. When glutathione depleted, the toxic metabolite binds to self-hydro-containing proteins in the liver cells or even in the kidneys causing cell death or toxic hepatitis. And that is the reason for the death or toxicity which is caused by paracetamol. So this is an N-acetyl parabenzoquinone. As we looked at it, it's a highly it's a strong electrophile which is formed through oxidation of paracetamol by the cytochrome P450 through the cytochrome P450 pathway. If you look again at your handout, you have that on page 19. I've given you the metabolism of paracetamol, which definitely explains or very clear on how it will cause toxicity. It is a treatment. You can give an acetylcysteine, which is thought to be a glutathione precursor, so it will increase your levels of the glutathione. It is also thought to be an antioxidant, therefore it will help to reduce the toxic metabolite into a safer metabolite, and therefore reduce it gives protection from toxicity. If you give it before 8 hours, it will give you, if you give it to the patient before 8 hours are over from time of ingestion of the very high doses, it can give complete protection. 8 to 24 hours to give in complete protection, but lower mortality. After 24 hours, shown to decrease mortality. After 24 hours, it has been shown to decrease mortality in established hepatotoxin, but very slight. You have very slight activity after 24 hours because there already will be the hepatotoxicity. But in case you still have some of the paracetamol not having bound the cellular tissues, it can decrease that. But preferably, it should be given before 8 hours are complete. Before 8 hours are complete, it has been to have very good activity. Another one is methionine, which is also an oral antidote. It can only be used if the patient presents within 8 hours post ingestion. Of course, is conscious, does not have this should be is conscious. Patient presents with 8 hours post ingestion, is conscious, does not have uncontrolled vomiting, and if activated charcoal has not been given. So we need to correct there. So because it's an oral antidote, it can only be given 8 hours post ingestion. The patient, of course, must be conscious to be able to swallow. If they have uncontrolled vomiting, they just vomit it out. And if you have activated charcoal, the activated charcoal, remember, will absorb the drug, and therefore the drug will not have any activity. N-acetylcysteine is a drug of choice for the treatment of an acetaminophen overdose. It is thought to provide cysteine for glutathione synthesis and possibly to form an adduct directly N-acetylparabenzoquinone, because we have said the cysteine, the N-acetylcysteine can bind. It's an antioxidant. Being an antioxidant, then it can act directly on the to bind the N-acetylparabenzoquinone and therefore reduce it, and hence reduce its toxicity. There could be other methods, but those are the main ones which have been known. There could be other methods in which it could act, but those are the two main ones where it acts as a precursor for synthesis of glutathione. Remember, the more glutathione you have in the stores, the better, because the glutathione will bind the N-acetyl benzoquinone imine, and therefore, of course, once it binds it, it will be able to reduce it to a non-toxic metabolite, so that, and therefore reduce any possible toxicity. That is paracetamol, a good drug, but also a drug that can be very, very toxic if taken in the wrong dosage. Paracetamol is a poor inhibitor of prostaglandin synthesis in the peripheral tissues, but it's more active on COX in the brain. Say that we do have 3 isoforms of COX, the COX enzyme, COX1, COX2, and COX3, which is found in the brain. The ability of paracetamol to inhibit COX3, an isoform so far located in the dog brain, could also account for its analgesic and antipyrectic action. it has no effects on the CNS. I think that is the end of our paracetamol. That is the end. So we are saying it looks like, of course, paracetamol will have its unique way of activity, because as we have said, it does not meet the SAR1, where it really does not have an acidic center, but it is still able to bring about antipyretic, analgesic activity, but not anti-inflammatory activity. We have questions? So we are okay. We are satisfied. Unfortunately, we are still lagging a bit behind. So now you should be ready for the first cut. We should include all that we have done about on the non-steroidal anti-inflammatories. So that today's lecture, that should be the end. Apart from the little bit I can remember, I think I should be able to come with it to put it next time. There was a small bit of the SAR of the prostaglandins. I remember that I didn't do. So that bit I'll go back. But only that small bit. Are there any questions? And if there are no questions, as I said, our topics are usually very broad. So we are done with the non-steroidal anti-inflammatories. And from the non-steroidal anti-inflammatories, the next subtopic is the antihistamines. And when we look at the antihistamines, we have H1 antihistamine or H1 blockers and H2 blockers. So under the histamine and antihistamines, we look at H1 blockers, H2 blockers, and also we also look at the proton pump inhibitors. I hope there will be enough time. Look at the proton pump inhibitors. Why we look at the proton pump inhibitors, although they are not antihistamines, is because the H2 blockers are actually used for ulcers. So we would want to complete the whole story of the management of ulcers. So in that, I will include also the proton pump inhibitors. So we look at the histamine 1, histamine 2 blockers, and the proton pump inhibitors, so that we can complete that story on the ulcers. So if there are no questions, then why don't I introduce histamine and H1 antihistamines? We want, first of all, to understand histamine, because when we look at the antihistamines, they are blocking the histamine receptor. They are blocking the receptor where histamine binds. So for them to be able to block the histamine receptor, it's just the way we say it. For us to understand the non-steroidal anti-inflammatories, it is important for us to understand the prostaglandins. It is important to us the arachidonic acid, because these drugs, which we call the non-steroidal anti-inflammatories, must be going for the same receptor as that receptor which is bound by arachidonic. So there must be some comparison between arachidonic and non-steroidal anti-inflammatories. And the same case applies to the topic we are looking at. Antihistamines, they must have some similarity. They will have similarity and difference, because you can see they are anti. So they are not agonists. They are antagonists. But for them to bind the same receptor that is bound by histamine, we must have some structural resemblance and some structural differences that allows these drugs to be antihistamines or to be antagonists rather than agonists. So that's why we start with histamine, for us to understand histamine so that we can understand histamine is an endogenous compound. In understanding histamine, it was possible to develop drugs that can bind that histamine receptor, but not elicit, not trigger any pharmacological activity, and therefore be antagonists. And that's all we are talking about. That's why it is important for us to look at histamine. And the first question is, what is histamine? What is histamine? And we can note that histamine is a beta imidazole. So now we are introducing a new ring, a beta imidazole ethylamine hydrophilic molecule. Imidazole ethylamine hydrophilic molecule consisting of an imidazole ring, an amino group connected by 2 methylene groups. So that is one description. That is a structural description, which we are going to look at in details. It has a specific structure. It has a simple, specific structure. It has an imidazole ring. It has an ethylamine group. It has an ethyl and an ethylamine group. And of course, they are connected in a very specific manner. It has also its physiological definition. It is physiologically active. It's a physiologically active amine with that molecular formula found in plants and animal tissues and released from mast cells as part of an allergic reaction in humans. So when you are triggering some of the allergic reactions, you release some allergen. That allergen will prompt the degranulation of mast cells, and the degranulation of mast cells will release histamine. So the degranulation of mast cells will release histamine, and then histamine will go to its respective sites and bring about those reactions that you see. So we are saying it's a physiologically active amine found in plant and animal tissues and released from the mast cells as part of an allergic reaction. So remember, an allergic reaction more often than not involves the degranulation of mast cells. When they undergo, some of the things that come out of those mast cells is histamine, and then histamine goes to its receptor, H1 receptor, goes to its respective receptors and binds those receptors and brings around a bit about what we are calling the allergic reaction. What are some of the reaction it brings? It stimulates gastric secretion. So once the histamine is released, there will be gastric secretion. It will stimulate dilation of capillaries. Once it stimulates dilation of capillaries, that fluids will start to leak out. They will leak out. Constriction of bronchial smooth muscles, they constrict, an allergic reaction or anaphylactic shock or asthmatic-like reaction, decreased blood pressure. So these are some of the effects of the histamine. And therefore, even when you look at this histamine, it doesn't seem to have any positive effects, sincerely. It is stimulating gastric secretion, because gastric secretion might help us in terms of causes dilation of the capillaries, that definitely not positive. Constriction of the bronchial smooth muscles, not positive. Decreased blood pressure, not positive. And that is why sometimes when you have the antihistamines, they are able to block the histamine with minimal toxicity, because this molecule has minimal advantages in the body. Imidazole ring and the imidazole ring and the amine group. So there we are. This is now our imidazole group. That is our imidazole group. There, two nitrogen separated by a carbon. And then this is our ethylene moiety in our amino group. We are going to look at that structure later. Synthesis of histamine, as we have said, it occurs in our body. It is found the amino acid histidine in a decarboxylase enzyme catalyzed by the enzyme histidine decarboxylase, occurs primarily in the mast cells and the basophils. So that is our histidine. This is an amino acid. That's an amino acid, histidine, and then histidine decarboxylase, and you have your histamine. All that has happened is removal of the carboxyl group. Removal of that carboxyl group gives us histamine, and this is the compound that we are discussing at this moment. Histamine, which is released from histidine. Now, the histamine receptors, there are several histamine receptors. Our main interest will be on H1 and H2. As I have said, H1 for antihistamine, the normal or the conventional antihistamines, the H2 for the anti-ulcer drugs, and H3 and most recently H4. So we can have H1, H2, and H3 receptors. Most of them are G protein coupled receptors. We can note activation of H1 causes contraction of smooth muscles in the gut, the uterus, and the bronchi. Contraction of the bronchi leads to asthma. So once histamine binds the H1 receptors, It causes contraction of the smooth muscles in the gut and the uterus and the bronchi, and that leads to asthma. Stimulation of H1 receptors on smooth muscles in fine blood vessels causes muscle relaxation. So in different areas, this was in the smooth muscles of the gut. This is now muscles in fine blood vessels causes muscle relaxation, and the resulting vasodilatation may result in severe fall in blood pressure. Remember, we said histamine results in fall in blood pressure. So we are explaining how does it come about. Muscle relaxation and the resulting vasodilatation may result in severe fall in blood pressure. In the CNS, H1 and H2 receptors predominantly are localized on postsynaptic membranes. You will do more of that in pharmacology. The H3 receptors appear to function predominantly as presynaptic receptors, possibly as histamine autoreceptors, and also being detected in some peripheral organs. H4 receptors exhibit very restricted locations in intestinal tissue, spleen, immune system, T cells, and so on. So we will not be looking mainly at H3 and H4 receptors, but it's important to note that we have the various receptors. We have H1, H2, H3, H4, but the ones we are most interested in are the H1 and the H2, because as we have said, when you block the H1, you are able to block the allergic reaction or the allergic response. When you block the H2, you are able to block the gastric or the acid secretion. And in blocking the acid secretion, if somebody has an ulcer, then you'll be able to manage that ulcer because there will be lesser acid being produced. So our main area of interest will be the H1 and H2 receptors. But it's also important to note the other receptors. And as I have said, fortunately, we work as different teams. So the pharmacology team should be able to mention the receptors in much more details. Structural consideration. So we are looking at this is our histamine molecule. So this portion here is what we are calling the imidazole ring. That is our imidazole ring. Then this here, between here and there, that is an ethyl or ethyl side chain or ethylene side chain. And of course, that here is definitely a basic amino group. It is highly basic because you can see it's not attached to any aromatic ring, very, very highly basic. And therefore, this amino group will be more often than not protonated at physiological pH. Then from this amino group, this is the alpha carbon, that is the beta carbon, and that is where the name came from, where we called it, it's beta ethyl imidazole ethylamine, because the imidazole group is attached to the beta carbon. When you consider this ethylamine, it's attached to the beta carbon of the ethylamine, and no wonder the name where we called it a beta ethylamine derivative, beta imidazole ethylamine. It's beta. This beta is coming from the point of attachment of the imidazole ring, because the imidazole ring can be attached at different positions. This could be a CH3 and it can be attached here. If it's attached here, then that will be an alpha imidazole ethylamine, but it is attached at that position, and then no wonder the name beta. So again, when we look at the histamine, we will also find that these different nitrogens are given different names. This nitrogen here, the one which is more basic, the one which carries the proton, is called the telenitrogen, and this is the pros nitrogen. So this is the pros or pi nitrogen, and this is the telenitrogen. And as we move on, we will be able to see the importance of these two nitrogens in terms of binding the different receptors. And then, of course, that is our basic amine. Of course, that is our ethyl side chain. So basically that very simple molecule of histamine. And from that endogenous compound, we are able to develop, we are able to get our antihistamines, because as we look at for the histamine, the drugs that are of importance are definitely the antagonists, as I have said, and not the agonist. It contains, so when you look at the structural consideration, it contains an imidazole ring, an ethyl side chain, and a basic amine. That's just imidazole. It's just another with ring. Imidazole undergoes tautomerism. This is not ketoino tautomerism. This, of course, is a different type of tautomerism. And that tautomerism, all we are saying is this nitrogen here, this either of these nitrogens, this hydrogen can also shift. This can shift, this can shift, this can shift here, and then this shifts to this, and therefore the hydrogen is on the pros nitrogen or the pi nitrogen, and the double bond is on this one. So what we are saying is that we can have tautomerism, not ketoino this time, but tautomerism, where either of the nitrogens can carry the hydrogen. That is a tautomerism that we are talking about. But we are going to look at it more. It can undergo tautomerism. It is basic in nature because of the aliphatic amino group. It is available in different ionic species. We are going to look at that. We have the N-tele tautomer. This, I think I want us to look at this a bit later.